Good morning dear students. Today's topic is host-microbe interaction. Host-pathogen interaction. The skin and mucous membrane is an environment for microorganisms to interact with the body. The interaction between the host and organism is referred to as symbiosis, and the relationship between a host and pathogen is dynamic. So, pathogen, they get interacted through a host by means of their skin or mucous membrane, and the interactions are referred to as symbiosis. And the symbiosis relationship between host and pathogen is of three forms. First one is mutualism. Here both the partners get benefited. An example is the relationship between ruminants and bacteria that is living inside the rumen of the ruminants. Another example is the probiotic bacteria that is seen in association with our body. Then a second association is commensalism. Here one of the partner get benefited, but other partner is unaffected. So example is the normal flora that inhabit the ice. They thrive on the secretions and dead cells present on the ice, but they do not affect the host cell. And another association is parasitism. Microorganisms get benefited by this parasitic association, but humans or the host get highly affected. So example, parasite that is living in the gastrointestinal tract of human and animal. Mechanism of host-pathogen interaction. Pathogenesis is a method by which a disease can develop, and the mechanism of pathogenesis involve following steps. First one is transmission, then adherence, colonization, and pathogenicity. Pathogenicity involved two different stages, invasiveness and toxigenesis. So first one is transmission. The pathogen, they must find a transmission route in order to get into the host and start infection. Transmission is the process by which pathogen is gaining access to the host body. Transmission of an infectious agent can occur in many ways, either through an exposed skin such as a puncture or a wound in the skin, or through the mucous membranes present in the gastrointestinal tract, respiratory tract, or urinary tract. And also there is another way of entry, that is orophycal route. While we are ingesting some contaminated food or drinks, then also microorganisms can get into our body. So these are the various ways in which pathogens get transmitted into a human body. Next step is adherence. After the transmission of a pathogen, it gets attached itself to the host tissue. Attachment is otherwise called adherence, and it is the necessary step in pathogenicity. Microorganisms contain ligands, which are projections that attach to host receptors or host surface proteins. So eukaryotic cell, they contain, their surface possess certain receptors which are either carbohydrate or peptide residues, and these specific receptors, they can bind with the ligands that is present on the surface of the microbes. Bacterial ligand, it is otherwise called adhesion, and it is a macromolecular, adhesin. Adhesin is a macromolecular component present in the bacterial cell surface which interact with the host cell receptor. Receptor binding is usually specific, that is receptor binding with this bacterial ligand adhesin is highly or usually species specific. So in this adherence step, the surface receptors, which is either carbohydrate or peptide origin, present in the eukaryotic cell surface, interact with the ligand adhesin seen in the surface of the bacteria, and this interaction is highly specific. Adherence. Pathogens have specific adherence mechanisms to attach to the cells or tissue surface, and they include tissue tropism. Here pathogens prefer specific tissues. For example, the hepatitis virus prefers liver cells. Then pathogens that infect only certain species, that is species specificity. So pathogens, it will not affect all the animals or all the plants. Pathogens are highly species specific, so it will infect only a particular group of animals or a particular plant species. Then next is genetic specificity. That means certain surface mutations might occur to this pathogen so that the host antibodies cannot recognize the invading pathogen. So these are the different mechanisms for the adherence of a pathogen to a host body. Next is colonization. So after transmission and adherence, the microorganism have to colonize within the host cell. So the first stage of microbial infection is colonization. It is the multiplication of pathogenic organisms, and here in order to colonize within the host cell, microorganisms produces toxins. They produce toxins mainly to avoid the normal microbial flora which compete them for space and nutrients. And this normal microbial flora is also creating some unfavorable conditions for the growth of these pathogens. So pathogens usually colonize host tissue that are in contact with the external environment, and site of entry of this pathogen to the human host mainly include urinogenital tract, digestive tract and the respiratory tract. Then during colonization, the host began to show signs of septicemia, that is toxic shock. So toxin is produced, so it affects the normal microbial flora as well as the host cell. So for infection to proceed, an infectious dosage should be determined. That means a minimal number of microbes should be necessary to cause a or to establish an infection. So certain pathogens, they are less contagious. That means they are less virulent. So a large number is required to cause a infection. For example, Shigella, it is a highly pathogenic bacteria. 10 to 100 will be enough to cause a serious disease. But Salmonella, it is less toxic when compared to that of Shigella. It will be required in 10,000 or 1 crore cells will be required to produce a infection. So that is the that dosage, that the amount of microbe that is necessary to establish an infection, that dosage is called infectious dosage. Pathogenicity. So after colonization, after getting enough number, after multiplying by binary fission and attaining enough number for the infection dosage, it will result in pathogenicity. So pathogenicity, it is the ability of the microbe to cause or to initiate the disease. Pathogenicity in bacteria may be associated with the unique structural components of the cell like capsule, fimbriae, lipopolysaccharides or other cell wall components like S layer, glycocalyx, etc. So these all outer membrane layer helps the bacteria to establish the pathogenicity or to establish infection in the host cell. So active secretion of the substances by the pathogen that either damage host tissue or protect bacteria against host defenses. So bacteria or pathogen, it will secrete so many substances. It helps the pathogen to resist the defense of the host defense mechanism, or it helps the pathogen to damage the host tissue. So pathogenic bacteria cause a disease by invasiveness and toxigenesis. So first step is invasiveness. In invasiveness, it is the ability of the pathogen to invade tissue. So invasiveness, it is the ability of the pathogen to invade a particular tissue to cause the disease. So mechanism for colonization, that is adherence and initial multiplication, it has already been taken place in the host cell. Then production of extracellular substances, that is invasins, that promote immediate invasion of the tissues. And after colonization, it will produce certain extracellular substances called invasins, which helps the bacteria to invade tissues. Ability to bypass or overcome host defense mechanism which facilitate the actual invasive process. So it will this pathogen should have the ability to overcome the defensive mechanisms that will be shown by the host cell to get or to start the infection. Invasion. Microbes begin to invade the host and produce bacteremia, that is presence of bacteria in the blood, or viremia, the presence of virus in the bloodstream. So it will begin to invade the host cell, and microorganisms are exposed to many barriers within the host cell. That is, host will either produce antibodies against these microorganisms, or it will have phagocytic cells, or macrophages or neutrophil will engulf these microorganisms, or it will have complements, or T cell cytotoxic or helper T cells will cause the destruction of the pathogen. So the pathogen has to face all these barriers that are being created by the host cell. Certain strains of bacteria produces invasins that activate host cell cytoskeletal machinery, enabling bacterial cell to gain entry into the cell by phagocytosis. So bacteria itself produce certain compounds called invasins. So it will help the bacteria to gain entry into the host cell, host cytoskeletal machinery get activated, so that the bacteria will be engulfed by the phagocytic cells. Engulfment of the bacteria by the phagocytic cell normally will be helpful for the lysis of the pathogen, but in some cases the invasins will prevent the formation of phagolysosome complex, so that inside the phagocytic cell bacteria multiply by means of binary fission, and it will cause the entire destruction of the phagocytic cell, and it will produce a lot of bacterial progeny. So certain strains of bacteria, they produces invasins which allow the bacteria to be engulfed by phagocytosis. Some bacteria are able to cause disease while remaining in the epithelial cells itself, but some others have to penetrate deeper. So some bacteria, well, they form colony in the external environment, or that is the cells that are very close to the external environment. The bacteria form colonies, and here itself it can cause infections or diseases. But most of the bacteria, they have to enter or invade into the inside of the cell. They have to penetrate deep into the body to start an infection. So once the barrier has been penetrated, this pathogen can multiply without competition. Then next is toxigenesis. Toxigenesis is the ability to produce toxins. Toxic substance produced by bacteria may be transported by blood and lymph. It causes cytotoxic effect at the tissue remote from the original point of invasion or growth. So as the toxin is able to circulate through the blood and the lymph, it will create cytotoxic effect in different regions other than the original point of infection. Next is evasion of host defense. How this microbe is able to tolerate with the host defensive mechanism. After colonization, multiple mechanisms are used by the pathogens to evade a host immune system. It avoids phagocytic recognition by producing capsules which prevent phagocytosis. So some bacteria which hate or which does not want to get lysed by a phagocytic cell, it will produce certain capsules around the cell, thus prevent phagocytosis. Then certain produces membrane damaging toxins which kill the phagocytic cell. An example of toxin which kill the phagocytic cell is leucocidins. Then some pathogen interfere with the complement activation. Some can survive in the phagocytic cell. Some can produce protease enzyme which allow them to avoid antibodies. Some pathogens produces catalase which prevent digestion of the engulfed pathogen, and some utilizes antigenic variation to alterate the antigen structure. So by antigenic variation, they can alter their antigen structure so that antibodies cannot be formed against that. Pathogens, they have the ability to mimic host molecules which can cause disease related damage, that is autoimmune responses can be elevated. So such type of damages or such types of mechanisms will be shown by the microorganism to escape from the host immune responses. Next is diseases to host. So after toxigenesis, diseases will occur to the host either through direct or indirect pathway. Direct method produce toxin, which are poisonous substance that produce toxemia within the host. Three types of toxins are produced. Exotoxins. These cause damages to the host. Example, botulinum toxin produced by Clostridium botulinum. Then tetanus toxin, etc. Then endotoxins, they are released when the cell get killed. And exoenzymes, they are enzymes that function outside the host cell or tissue. So they include coagulase, which form fibrin coat that hide the microbe from phagocytosis. So this exoenzyme, example is coagulase. It will produce a fibrin coat that prevent the microorganism or that protect the microorganism from phagocytosis. Then hyaluronidase is another exoenzyme which break down connective tissues of the host cell. Then fibrinase, it helps to break down blood clots to allow pathogens to continue spreading. So these are the different exoenzymes that are produced by the microorganism for rapid infection in the host cell. So that's all about the infection. Then next is virulence. Virulence or pathogenicity is the ability of the microorganism to infect or damage a host. The degree of damage caused by a microbe to its host is called the virulence. Pathogenicity of the organism depends on its ability to cause disease, and it is determined by its virulence factor. So, virulence factor determines the pathogenicity of a microbe. And many virulence factors are effector proteins that are injected into the host cell by special secretion mechanism. An example of such a secretion mechanism is type 3 secretion system, which is seen in some type of bacteria. The virulence factors of bacteria are typically proteins or other molecules that are synthesized by enzymes. So, most of the virulence factors which are responsible for pathogenicity in bacteria is a protein which are synthesized by some enzymes. These proteins are coded by genes in chromosomal DNA in case of bacteria, bacteriophage DNA in case of virus or plasmid. So, these virulence factors which it will provide additional pathogenicity to microorganism, and this virulence is caused by these virulent factors, and these virulent factors are proteins which are mostly synthesized by some enzymes, and they are mostly coded by the genes that is present either in the bacterial chromosome or plasmid, and in case of bacteriophage it is the bacteriophage DNA. Then an example of virulence factor is the Helicobacter pylori, that is causing ulcers to human beings. It is capable of producing an enzyme called urease. So urease, it is a virulence factor which allow this Helicobacter pylori to invade the gastrointestinal tissues and cause ulcer there. So this is the host microbe interaction diagram. Here the first one is invasion of a host through primary barriers. So first the pathogen, it will invade the host through any of the primary barriers like skin, gastrointestinal, urinogenital, or any of the entryways. Then evasion of the host and tissue host defenses by the evasion of local and tissue host defenses by the microbe. So microbe has to escape from the host defense mechanism by various methods which we have already discussed. Then microbe replication with or without the spread in the body. Then host immunological ability to eliminate or control the microbe. At last, host should have some immunological ability to should develop some immunological ability, otherwise we have to administrate some drugs to eliminate this pathogen. So these are all about the host microbe interaction, and these are some of the important references that I have made in order to prepare this note, and I have also searched Wikipedia. So this is all for today's class. Thank you.